It's Mr. Dunbrook. Hope you guys are doing well. Coming to you live from my basement, unless you're watching this later. Just wanted to take a couple of minutes to ask you how you're doing and make sure you're doing well. And please make sure you're commenting and posting on Google Classroom. I'd like to hear from you guys. A lot of you are doing a really good job as far as taking care of your assignments and getting them in on time goes. So thank you very much for that. What I'm going to do on this video is I'm going to go through the PowerPoint for Chapter 11, and a little bit of this first part of the PowerPoint is going to be review for you, so you can go through this first segment very quickly. What I'm going to try to do is chop this into roughly 20-minute segments and post those each on YouTube in different parts or different segments. So again, I hope you guys are doing well. And please reach out to me with any questions or any concerns or any issues you might be having. So I'm here to help, and let's get through this together. Okay, I'm going to click off my my mug shot here, and let's get to genetics. Again, bye. If this is your first go-around through this PowerPoint, please maybe slow down the video, pause it when you need to, do whatever you have to do in order to kind of start absorbing some of this stuff. You also have other resources such as your textbook, Chapter 11, and so on. This PowerPoint you should have in front of you right now. You have a paper copy of it. If for some reason you lost your paper copy, it's also down in the stream. It's kind of far down now because this chapter goes back a little bit into the past. So it is posted, though, it's kind of far down in the stream, Chapter 11 Genetics PowerPoint. So I'll give you a minute to grab that. Actually, just pause the video and grab that, and let's get started. So hopefully you have this in front of you as well as something to write with. Again, this first segment I'm going to go through quickly because we already went through this in class, but I want to get your brain thinking back from the beginning because this builds upon itself. Okay, so Chapter 11, Genetics. Genetics. All right, first of all, you guys know that we can't talk about genetics without talking about a lot of vocabulary, things like offspring. The word offspring literally means the children of sexually reproducing parents. So if you are a plant or an animal or even a mushroom, sometimes a fungus, you will find that if you combine different sex cells from two different parents, typically male and female, but there are some strange exceptions out there in nature, you will find that you get genetically different offspring, genetically different children. And this is because they get their genetic information from their parents. So obviously they're going to kind of resemble their parents in certain aspects. However, you can be carrying traits, you can be showing traits that your parents do not show, but that they carry. Keep that in mind. Again, we discussed this in class. All right. This whole concept is known as heredity. You kind of see the word inheritance in there, the root word H-E-R-E-D, the hered. It's a set of characteristics an organism receives from his parents, heredity. Again, vocabulary rich in this section, so make sure that you are kind of making sure you master this vocabulary. You guys did your vocabulary assignment yesterday, and most of you have turned it in. Those that haven't, please get to it now. So this whole story really begins in Austria with a guy named Gregor Mendel. He was studying in the 1800s in the country of Austria, which kind of borders Germany and the old Czech Republic and Switzerland. He was a monk, and a monk is somebody who's studying to be a priest. So it all started with this Gregor Mendel guy from the Czech Republic and Austria. In 1822, he was born. Austrian monk, not Australian, Austrian monk. We talked a little bit in class about how he is not that monk from the TV show. Also, he had a lot of time on his hands. He had a lot of time when he wasn't studying the Bible and copying the Bible into printed messages for the masses. What he did was he started a garden. He started growing pea plants, and he started noticing some interesting things about the characteristics or traits of his pea plants. I will remind you, pea plants reproduce sexually. People still are having a hard time believing that plants really reproduce sexually. They absolutely do. It takes a mama and a daddy plant to make a seed in a baby plant. So a baby plant really comes from a seed. A seed is a baby that has been produced by not sperm and egg, but pollen and egg, which is the equivalent to sperm in plants. So plants absolutely reproduce sexually. Can you grow a plant asexually? Well, yeah, you can. You can take a clipping off of a branch and stick it in soil, and you can grow a clone. That would be a way of reproducing the plant, and that would be asexual. But in nature, that doesn't happen very often. Typically, it's literally the birds and the bees pollinate, and we get sexually reproducing organisms, or sorry, sexually producing results of our offspring, and they are typically seeds that will be found in fruits and inside the flowers of plants. So again, big picture here. The pollen of a plant is equivalent to the sperm of an animal. Same thing. These are gametes. These are 50% the genetics of each parent. So just like a male sperm for a human would have 50% DNA, and an egg from a human female would have 50%, same with a plant. 50% genetics in the, or DNA in the pollen, which is the male sex cell, 50% DNA in the egg or ovum, which is the female sex cell in a plant. And when those combine, that is sex, technically, and you get a seed developing. And typically that seed develops in a flower or a fruit or sometimes a cone, okay? And it will, the seed will then have 100% of the DNA, of course. So again, quick anatomy of plants. Typically plants and flowers in general, I'm going to give you a real generic version here, will have male parts and female parts, just like animals. The anther is this whole entire thing is called a stamen. It's divided into the anther and the filament. This is equivalent to the male reproductive parts. The stamens are the male reproductive parts. They include the filament and the anther. These produce sperm. This is, really analogous or identical to an animal's testes. The female parts of the female flower produce the egg or ovule. Again, this is equivalent to maybe a female animal's ovaries, okay? These are plants, but they reproduce sexually. And because of the fact they reproduce sexually, 50% comes from mom, 50% of the DNA comes from dad, they follow the same rules as humans. So if you're thinking, well, why do I care? Gregor Mendel studied, genetics in plants. Well, because the same rules apply to us, because we reproduce the same way, really. And I know that's hard to believe. All right, so that's not good. Let me pause this for a second. See what's going on with my failure to load here. Okay, we're back. Sorry about that glitch. So again, big picture, plants reproduce sexually, and therefore they follow the same rules as far as genetics and inheritance goes as a human would, or any other animal for that matter, or any other sexually reproducing organism for that matter. All right, so plants can reproduce by self-pollination or cross-pollination. Now this is a little different than animals and especially people. We can have plants that have both male and female parts within the same plant. So for instance, this one flower here has these female stamens and anthers, and these male stigma and style and ovaries. Don't worry about those names right now. But some plants can actually absolutely pollinate themselves. Believe it or not, this is still sexual reproduction, because all of the sperm or pollen that came from this male part right here, and I'm going to try to highlight on here, that came from this male part right here, and all of the female eggs that would be in what's equivalent to the uterus, or ovaries, I should say, of this plant right here, are each genetically different. They have variety in them. So we talked about how variety is achieved through meiosis when you guys did your meiosis cards that you had to put in order, and you saw that every time you make a sperm or egg cell, they are always different from each other, which is absolutely not the case for mitosis, where all cells that came from mitosis were genetically identical. You can also have cross-pollination, and this is more kind of comparable to an animal, where one plant is a male plant and he has pollen, or equivalent to sperm, and another plant is a female plant and she has only flowers that have ovaries with eggs in them, or ovum, and that's kind of traditional sex, where one organism that's male gives gametes or sex cells to another organism that is female, and the female would end up carrying the seeds and therefore have the baby. Remember, 50% or haploid pollen or sperm, male gametes, would meet up with 50% egg, 50% DNA in that egg, I should say, haploid gamete, and the combination of the dad's sex cell and the mom's sex cell would give you a baby that is 100% diploid. We've said this many, many times now. Okay, here we go, continuing on. And Mendel had a lot of time on his hands, as we said, and he worked in this beautiful monastery in Austria. You can still visit there today if you want to geek out and go take a virtual tour. All right, true breeding. We've gotten into these concepts already. Some more vocabulary coming at you. Think of this as the same as a purebred. If you're using true breeding organisms, as Mendel was, every time you have babies, you can expect the same traits. Much like every time you have, purebred pit bulls, they always look the same, or purebred Dalmatians, they always look the same. They have the same traits. So that was important that Mendel started with these true breeding types of organisms, because you have to know what you're starting with in order to be able to make sense of your results. So he went to great pains to make sure that he knew what was breeding in multiplying or crossing with what. So he actually would remove female and male parts of flowers to keep them from reproducing together if he didn't want them to. And then he would actually take male parts and spread them to female parts to control what type of plants were actually reproducing. Very, very important. Also keep in mind, they have to be the same species in order to reproduce sexually. So even though this is a white flower and this is a purple flower, these are both still flowers from pea plants. They're just different varieties, just like we have varieties within the human species, and we can still reproduce. That's what sexual reproduction gives you. It gives you variety in a species. So as we've said all year long, because we're all diverse, what kills one will not kill all. That's a beautiful thing about diversity and variety, and that's caused by sexual reproduction. So when he cross-pollinated these two different types of flowers, these two varieties of pea plants, I should say, he would get these seeds, and these are peas. You guys have heard of peas in a pod. These are the same as brothers and sisters in a mother's womb. I know that's crazy, but that's what it is. And again, half the DNA from one plant, half the DNA from another plant. All of these babies are brothers and sisters, and they are all genetically different. When he planted those peas, remember, peas are really seeds, and when he planted them, this was a big deal. He took a purple times a white, or crossed with a white. And if you're Mendel, if you're the 1800s, I want you to think about what you might expect the result to be for that. Would you expect it to be purplish plants, white plants, purplish whitish, like a lavender? Would you expect it to be purple and white striped? Would you expect it to be, I don't know, maybe some purple, some white? Well, he was hypothesizing all these different things, and lo and behold, the F1 generation, remember, here's our P generation, we've talked about this before, the first set of children, the F1 generation, lo and behold, all purple. And Mendel said to himself, what the heck is going on here? He was expecting not that, anything but that. So that got him kind of thinking. He said, why is this happening? And that's typically good science comes about from surprises, and we want to know more. So chapter 11, Mendel continued here. The traits that Mendel used in his experiment. You guys can see these. They might be a little difficult for you to see the smaller words, but he used traits such as the shape of the seed, the color of the seed, the shape of the pod, the color of the pod, the flower color, the flower location, whether it was tall up on the tips of the branches or down in what we call the axils or nodes of the branches. Also the plant size, small or really tall or short. And each trait had two different varieties. This is very simple genetics here. Each trait had only two different varieties. That made life very easy for him. Of course, as you know, there are many traits that have way more than two varieties. Think of eye color. Think of even skin color has about 8 different varieties, believe it or not. Okay, so to maintain these true breeding populations of plants, remember, Mendel didn't want anything kind of cross-contaminating his plants, so he put them all in a greenhouse to keep out the birds and the bees from cross-pollinating things that he did not want to have cross-pollinated, okay? So as we said earlier, he would create a situation where he could control what plants were pollinating what plants, and he would do this by cutting off male parts on some plants and then cutting off female parts on other plants, and so on and so on. If you cut off the parts of male parts, the male cannot get another female pregnant, and if you cut off the female parts, that female cannot get pregnant. All right, so let's look at this business of traits that Mendel was looking at. He was looking at things like seed color and seed shape and tall versus short. But here's some traits that you can look at for yourself. We saw this in the lab that we did, the Build a Baby lab. Eighth period, you guys did not get to do that. I apologize. Widow's peak. You can look at yourself in the mirror, see if you might have that. When you hold your fingers up, do your fingers kind of bow outwards like that, or do they come kind of straight up like that? Your thumb, are you double jointed? Your earlobes, are you attached earlobe or unattached earlobe? Can you curl your tongue? Believe it or not, that's genetic. You cannot learn how to do that. What color your eyes? What color your hair is your hair, and so on and so forth. Now, keep in mind, most of our phenotypes, that's what we physically show, are the result of our genetics, of our genotype, but also the result of our environment. However, some things don't have much to do with environment. For instance, if you can curl your tongue or not, that's really 100% genetic. That's not something that can be learned if you don't have the genetic trait that allows your tongue to curl. But a lot of other genes are very influenced by environment as well. All right. So here again are each of the seven traits that Mendel studied. Each had only 2 varieties, and Mendel got kind of lucky on that, because if they would have had 3 or 4 varieties for each trait, that would have made his work a lot more complicated. Again, a look at the 7 traits. Okay. His first parent plants of different varieties. Again, these are the same species. These are both pea plants. He called these the pea generation. So the pea is always going to be the original parents. And when they cross, the children are known as the F1. And as you cross F1 children together with each other, you get grandchildren, which would be known as the F2. When Mendel first did this, he took a tall plant versus a short plant, and lo and behold, he got all tall plants, just like with purple in white color. When he took white times purple, he got all purple. So this is where he's starting to kind of think of this concept of dominance in his head. Why weren't these plants medium? Why weren't some of them tall, some of them short? He's starting to think about these things. And again, the surprise is what led to the science. When he started off, these were true breeds or purebreds. These always produced tall plants if they were bred together. These always produced short plants when they were bred together. So we would call these pure breeds or true breeds. However, when he breeds one purebred with another kind of purebred, he gets what we call hybrids. And hybrids are this generation right here. Even though you don't know it, they are a cross between a tall and a short. You know it now, but he didn't really realize that yet until the second generation, the F2 generation, the grandkids. What do you see happening? Again, go back to the beginning. A tall versus a short, or combined with a short, I should say, reproduced with a short. First generation, 100% tall plants. But when I breed these together, I get mostly tall, but I get one short. We could also say that this short trait skipped a generation. It did not show up in this generation, but lo and behold, here it comes back in this generation. This also got Mendel thinking, what's going on? All right. So when Mendel did this cross, he thought that the hybrids would be a blend of both traits. As we already said, he was shocked to find that it wasn't the case. And you guys already know how to set up Punnets, but as one final reminder here, take the parent generation. Remember, every trait you have is a combination of 2 alleles, one from mom and one from dad, 2 different varieties, or sometimes 2 of the same variety from each parent. Remember, mom's egg and dad's sperm give you 1 of each allele for each trait, 1 of each variety for each trait. So when you see this big S, that is a combination of who this parent's parents were. When you see this little s, that is a combination of who that parent's parents were. And these are the children, right? So now if these children go to make children, they're only going to give 50% of their DNA. Notice there's only 1 S there, a small, and 50% over here, only 1 S, a large S, to their children. And that will go back up to the 100% DNA, what we call diploid. So you had diploid parents that made haploid gametes, sperm and egg cell, or pollen and egg in this case. And when we join the the haploid gametes we're back up to diploid children. Now we can breed those children together and make another Punnett square and another cross, and these would be the F2 generation. This would be the grandkids of the originals, okay? And again, as you go to the outside of the Punnett square, one allele for each trait from the dad, that's sperm cells, boom and boom, right there. One allele in each egg cell from the mom, boom and boom, because the mom's a diploid organism, but her eggs are only going to be haploid, so they'll only have one of each, or one of whatever she has to, half of whatever she has to give, I should say, 50%. And the same with the dad. 50% of what he is goes in his sperm cells. Half of his sperm cells would get a big S, the other half would get a small s, okay? And a couple more slides here, and then we're going to end this session, and I will post part 2 of the PowerPoint at a later date, probably tomorrow. In approximately 1/4 of the offspring from the F2 generation, the trait disappeared and reappeared. That's what we talked about up here, about this skipping a generation business. You didn't see any short in this F1 generation, and lo and behold, that short gene comes back, or that, in this case, I think this was wrinkled instead of smooth or something. I forget what the S stood for here. But we saw that homozygous dominant times a homozygous recessive, those would be the purebreds. The first generation or F1 children were all hybrids. They were kind of mutts. And of course, the second F2 generation, the grandkids, are definitely mutts or hybrids. But we see that that trait for the homozygous recessive, in this case, I think it was wrinkle, came back. It skipped a generation, and it came back in a 1/4 ratio. That is very important, a 1/4 ratio. And these ratios are what Mendel started really looking at. He saw patterns developing. Another example of that, P generation, green times yellow. Again, these would be purebreds and purebreds. So homozygous dominant and homozygous recessive or something. F1 generation, all green. So we're seeing, well, what happened to the yellow trait? Well, it comes back in that F2 generation. And not only does it come back, but it comes back at about a 3-to-1 ratio. In Mendel's numbers there, it was 428 to 152. But if you divide that out, it becomes very close to 3-to-1. And he kept noticing these patterns over and over again for different traits, which is what made him coin the term dominant and recessive. He said, well, this green must be dominant because it's kind of hiding the yellow in the first generation. But the second generation, the F2 grandkids, you see that reappear. So that must not have totally gone away. It must have just been hidden in that last generation, and now it has returned. But not only has it returned, it's returned only 1/4 times. Please keep in mind, Punnett squares are probability and prediction. They are not necessarily always reality. This data, however, is reality. This is direct observation from Mendel's gardens. Okay? All right. Scrolling on down. He started to coin some terms like dominant and recessive. He also started to coin the term Merkmol, which today we interpret as gene. So Mendel concluded that for each of the 7 traits that he looked at, remember he looked at color and size and peak pod wrinkliness or smoothness, all these traits he looked at, he said there must be a gene for each possible trait. And since plants are sexual, each plant must have 2 versions. This is very important, that whole diploid thing. Varieties of each gene, one from the DNA of the mom and one from the DNA of the dad. Okay, I'm going to stop right there. As soon as I talk this slide, remember from your meiosis cards, again, that the cards that you sequenced, genes are like pages in a chapter. Chromosomes are like chapters. So remember, you have 2 of each chromosome for a total of, if you're a human being, 46 total chromosomes, but really it's 23 pairs of chromosomes because you get 1 of each chromosome from each parent. So think of this as a book with 46 chapters, but you have 2 chapter 1s, 2 chapter 2s, 2 chapter 3s, and so on, because your mom gives you 1 of each of your chapters, and so does your dad. So that's going to conclude for us today, and I will post this on YouTube, and then I will post part 2. All right, everybody stay well.